Sixth year biology students sitting your leaving cert in June 2026. This is the final video of protein synthesis that you need to know. We have previously covered transcription, where we talked about DNA forming mRNA, mRNA carrying the code out of the nucleus, and now we are talking about the second part of protein synthesis, which is known as translation. Remember, we have mRNA is single-stranded, it's carrying the code to make the protein, and now we're going to talk about where that's going to go. So we have the organelle where proteins are made called the ribosome. The ribosome is made up of another type of RNA called rRNA, which stands for ribosomal RNA. So the single-stranded mRNA is going to bond with the ribosome. Now remember, we have groups of 3 bases called codons along the mRNA. Each codon codes for one particular amino acid, and there are over 20 common amino acids like serine, proline, lysine, valine, that we don't need to know the name of, but we do need to know that there are 20 common amino acids. Depending on the order in which those amino acids combine will determine what type of protein we make, because we know we have structural proteins, keratin, myosin, collagen. We have globular proteins like enzymes or hormones. So what determines those is the sequence of amino acids and also the folding. So what happens is mRNA bonds to the ribosome, and we can see the single-stranded mRNA. Now, 3 bases known as the codons on mRNA are going to code for particular amino acids. Now, how does the amino acid get into the ribosome? There is our final type of RNA called tRNA. Transfer RNA is going to transfer or carry the correct amino acid in which the codon on the mRNA has coded for. It's going to come in with its 3 bases known as an anticodon. Those 3 bases are complementary to mRNA's bases, and it's going to carry the correct amino acid in. And then what's going to happen is the ribosome moves along the mRNA, and the next codon codes for amino acid proline, which will come in and bond on, and then this is going to continue. The ribosome is going to move along the mRNA. Now what happens is when the tRNA has come in, brought the amino acid, eventually the tRNA detaches and the tRNA leaves, leaving its amino acid behind. The sequence of amino acids are held together or joined together by peptide bonds, and when the stop codon is reached in the ribosome, what will happen is that sequence of amino acids will be folded in order to form a functioning protein, and it will be folded into 3D globular shapes or folded into long fibers, which will determine the function of the protein. We can have globular proteins or we can have fibrous proteins, and the folding will determine the protein and the function of it. Globular being the likes of enzymes and hormones, or fibrous being the likes of myosin or keratin or collagen. And that process of the mRNA coming into the ribosome to make the protein from the sequence of amino acids is known as translation. So protein synthesis, the anabolic reaction, which starts off with transcription, then translation, and your 3 types of RNA, M, messenger RNA, R, ribosomal RNA, and T, transfer RNA. All have different functions, so make sure you know and get it correct in your head the order in which protein synthesis happens if you're a 6th year sitting your Leaving Cert Biology exam in June 2026.